In the last lecture, I told you about translation initiation in prokaryotes. Now, in this lecture, we'll be discussing about translation elongation in prokaryotes. So, initiation is complete when we get this complex. And I told you this complex is known as a 70S initiation complex. Now, what we have here is we have the 30S ribosomal subunit. We have the attached messenger RNA. We have the attached large ribosomal subunit, that is 50S ribosomal subunit. We have the initiator tRNA attached to the P site to the initiation codon. What we have free here is the amino acyl site or the A site, and we have the exit site which is free. So translation elongation begins with the formation of the 70S initiation complex. Translation elongation can be described as an elongation cycle or a reaction cycle where the amino acyl transfer RNA molecules are brought and bound to the A site of the ribosome. The amino acid present on the tRNA of the P site and the amino acid present on the tRNA of the A site then join together with the help of a peptide bond. So the second step in translation elongation is the formation of a peptide bond. Once the two amino acids are joined together with the help of peptide bond, movement takes place, and that movement is known as translocation. So there are three steps of elongation. First step is the binding of the amino acyl tRNA to the A site. Second step is formation of peptide bond by the peptidyl transferase enzyme. And third is translocation. What all do we require for the translation elongation step? We require the initiation complex, that is the 70S initiation complex in prokaryotes. We require all the charged transfer RNA, the amino acyl transfer RNA molecules to which amino acid has been attached, and they will be brought to the A site. We require elongation factors. There are three elongation factors which are required. We have EFTU, EFTS, and EFG. And we require energy, and that energy is in the form of GTP hydrolysis. This is the elongation cycle. We call it the elongation cycle. We call it the reaction cycle. We also call it the EFTU, EFTS cycle. So I'll start from here. We have a molecule of EFTU joined to a molecule of GTP that will provide the energy. And this EFTU GTP binary complex then searches for the amino acyl transfer RNA. So a ternary complex is formed. So the first step is the formation of a ternary complex which has three components: elongation factor TU, GTP, and the amino acyl transfer RNA, which has to be taken to the A site of the ribosome. So this ternary complex formation is then leading to translocation to the A site of the ribosome. So once it reaches the A site of the ribosome, it places the amino acyl transfer RNA to the A site and GTP gets hydrolyzed. So what we get is amino acyl transfer RNA attached to the A site, EFTU releasing with GDP. Now EFTU, this is an elongation factor that has to again attach to the next amino acyl transfer RNA molecule. So it again requires a molecule of GTP. So it has to exchange its GDP for another molecule of GTP. And here the second elongation factor comes in, which actually acts as a exchange factor, and that is EFTS. So EFTS now forms a complex with EFTU and GDP is released. So we have the EFTU GDP complex. EFTS exchanges GDP and attaches to the EFTU. So we have now EFTU EFTS complex which is formed. So this step is EFTU EFTS complex formation. Now this EFTS exchanges itself for a molecule of GTP. So EFTS will be released and we get EFTU and GTP. So this EFTU GTP, they will again form a binary complex, again start looking for the amino acyl transfer RNA, form a ternary complex, again take the transfer RNA to the A site, GTP gets hydrolyzed, these two factors are released, GDP is exchanged again for a molecule of GTP, and this cycle will continue throughout the process of elongation. The cycle is known as elongation cycle. We call it the reaction cycle. We call it the EFTU EFTS cycle. So EFTU, as I've described, is required to deliver the amino acyl transfer RNA to the A site. Energy is required, and that energy comes from hydrolysis of GTP. The released is EFTU GDP complex, which has to be regenerated. That is, EFTU GTP is required back to bind to another amino acyl transfer RNA. So for this regeneration, we require the elongation factor, that is EFTS. So it forms a complex EFTU EFTS, and this EFTU EFTS exchange cycle, EFTS displaces the GDP and get replaced itself by a molecule of GTP to again give us the complex, that is EFTU GTP complex, which binds to another amino acyl transfer RNA and delivers it to the A site of the ribosome. So FMET tRNA, that was the initiator tRNA, is bound to the P site attached to the initiation codon. Next codon, which is positioned in the A site, as shown here, the codon is GAA, and its corresponding anticodon, that is CUU. The tRNA brings in the second amino acid of the chain with the help of elongation factors. So appropriate amino acyl tRNA having a corresponding anticodon to the codons present in the A site is brought to the A site and is bound to the A site with the help of the elongation factors. We have EFTU and we have EFTS. So now what we have is that two amino acyl tRNAs are positioned, one in the P site and one in the A site. So the next step is a peptide bond will be formed between the two amino acids. The amino acid which is present on the P site tRNA is always donated to the amino acid on the A site tRNA. Therefore, the P site is also known as a donor site and the A site is also known as a acceptor site. So the bond between the amino acid and the transfer RNA at the P site is broken. This amino acid is transferred to the A site amino acid. So now what we have here is that the A site amino acid will be accepting the amino acid from the P site transfer RNA. The enzyme responsible for formation of the peptide bond between the two amino acids is the peptidyl transferase enzyme. And this activity, this enzymatic activity, is present in the large ribosomal subunit. So the peptidyl transferase activity of the 50S ribosomal subunit forms a peptide bond between the two amino acids, that is, the one transferred from the P site to the one that is present on the A site. So this picture shows peptide bond formation catalyzed by the enzyme peptidyl transferase. The activity is present in the large ribosomal subunit. Here we can see the polypeptide chain is extending. At the end terminus is always a first amino acid. That is, here in this case, it will be formyl methionine. Attached to the second amino acid, third amino acid, fourth amino acid, fifth amino acid, and the sixth amino acid. And the incoming seventh transfer RNA bringing the seventh amino acid. So the peptide bond is formed between amino acids. The polypeptide chain keeps on extending. And once this step is completed, then we move on to the third step of elongation, and that is known as translocation. So what happens here is we have the A site transfer RNA, which has accepted the amino acids. P site transfer RNA has donated its amino acid. So now P site does not have any amino acid attached. It's a deacylated transfer RNA. A site transfer RNA now has two amino acids attached to each other with the help of a peptide bond. So it's a dipeptidyl transfer RNA. Now the ribosome will move downstream, that is towards the 3' end, 3 places, so that the next 3 nucleotides come into the A site, or the next codon comes into the A site. So the transfer RNA now, which is present in the P site, will be moving to the E site. The transfer RNA, which has 2 amino acids attached and is currently in the A site, will move to the P site, and the A site will again become free for the next elongation cycle. So this is what is here. This was an initiator tRNA shown in blue. This was an initiation codon AUG, which was present in the P site. Now we can see it's present in the E site. So this is because of the movement. This arrow shows that the ribosome is moving downstream 3 steps, so that the next codon comes into the A site. Codons which were originally in the A site are now in the P site along with the tRNA, and that tRNA has 2 amino acids attached. The tRNA that was originally in the P site is now in the E site. This is an initiation codon AUG, and this movement is known as translocation. Now, translocation also requires a factor, and that factor is known as elongation factor G. We also call it translocase, and the movement requires energy in the form of GTP hydrolysis. So a complex of elongation factor G and GTP bind to the ribosome, and in an energy consuming step, the discharged tRNA is ejected from the P site. This is a tRNA which has donated its amino acid. The peptidyl tRNA, that is, which is present on the A site, it moves to the P site. GDP and the elongation factor G will be released, and new codon is now in the A site, which is again ready to accept the next transfer RNA, which is brought by the elongation factors. So this cycle will continue till any one of the stop codons reach the A site. So how to summarize the process of elongation? The first thing we had was 70S initiation complex, which had an occupied P site. The A site was empty. In the A site, amino acyl transfer RNA is brought. with the help of elongation factors, and energy is required, followed by the formation of a peptide bond. The peptide bond is formed, catalyzed by the enzyme peptidyl transferase. The P side transfer RNA, which has donated its amino acid, has no amino acid now. The A side tRNA, which has accepted the amino acid, has got two amino acids now, followed by translocation, where ribosome moves down on the messenger RNA. The transfer RNA, originally that was on the P side, now you can see it's on the E side, from where it will exit. The transfer RNA, which was originally on the A side, is now on the P side with both its amino acid. And as we can see on the N terminus, it's always the first amino acid. That is formylmethionine in case of bacteria. A site again is now free with its codon, and its corresponding transfer RNA will be brought here and will be attached to the A site. This is how elongation is taking place. It keeps on going till any one of the three stop codons reach the A site. So till then, the elongation cycle will go on. Corresponding tRNAs are brought to the A site by the elongation factors. Peptide bond formation takes place. Translocation takes place. The transfer RNAs from the E sites are released. Transfer RNA from the P site shift to the E site. A site again becomes empty, and the next tRNA is brought and attached to the A site. And this is how elongation takes place. In the next lecture, we'll be discussing about termination of translation. Till then, thank you.